with that, actually, I think I should just now hand over to Tabby. She has some questions for you. Tabby, you're on air.
Hang on, let me unmute on my end. Can you hear me?
Yes.
Yep.
Okay.
Please ask your questions. Yeah.
Once you live in the blindness world and raising kids with low vision, I think we know what the term blindness is and how that's defined. We live in kind of this space where blindness and low vision is kind of like a spectrum. Can Dr. Maguire or Dr. Bennett, either one, put a little bit of definition, put a little bit of description around what it means to be legally blind?
So legally blind means two different things, essentially. There's visual acuity, which everybody is very familiar with. That's reading an eye chart. And there's 20/20 vision. I think of it as a fraction. 20 divided by 20 is 100% normal central vision, whereas 20 over 200, which is the threshold for legal blindness, that would be 10%. Basically 20 divided by 200, 10% of normal central vision, and that's the threshold you cross, and that's considered legally blind for center vision. What's interesting is there are numerous retinal conditions, forms of retinitis pigmentosa and inherited retinal diseases, where people have 20/20 vision and they're legally blind, because having good center vision is very important, but that represents only a tiny fraction, less than 4% of your entire visual field. So people are essentially with that sort of situation are looking through a long tube or a gun barrel. If your peripheral vision is constricted within 10 degrees of center, which is a little bit harder a concept to understand. Sort of, it's not like visual acuity. It's a more abstract test. But if it's constricted within 10 degrees of the center, the normal being out to about 70 or 80 degrees, then you're so constricted that it impairs your ability to walk around and be mobile, to drive, to even do things like reading, and that's considered also a criteria for legal blindness. So there's visual acuity, which is 2200, and visual field, which is within 10 degrees, constricted down to 10 degrees.
Now, is it the blindness or lack of vision that qualifies people to come into the study? So I know there was some mention earlier about having functional retinas. So working with a lot of the parents and a lot of the kids that are in the LCA community, but also with other genetic disorders that affect vision, one of the big questions that we hear a lot is if kids or adults are already blind, can they still be treated?
So it depends on the mechanism of the condition, and I'll just comment on this RPE 65. So the cells, the nerve cells are still there, but they lack the molecule to capture light. So it's like having a car and with no gas in it, and it's. I wouldn't say it's completely unique, but it's a very unique situation. A lot of forms of blindness. It's not just the cells aren't working correctly, but they're actually degenerated. They're worn out and they're not there anymore. And that would require a different form of treatment than gene therapy, because there are no cells to treat. I think maybe Dr. Bennett could comment on other forms of how the biology works to cause the vision impairment.
Yeah, I would like to add that vision also depends upon the wiring being intact between the retina and the brain. And we don't know a huge amount about how long that wiring persists if somebody's retina is not functioning. There have been a number of very historic experiments studying this phenomena in newborn animals. But what we found with the RP65 was actually surprising, that even though the wiring between the retina and the brain hadn't been used for as long as 35, 40 years in one person, when we could treat the retina, we could activate that wiring and allow it to provide the information to the brain, which, of course, is what we use to have vision. So we don't know the answer for the other conditions, whether the wiring is still viable, but we certainly have optimism based on our findings with the RP65. And so, for example, if there is somebody with LCA who only has light perception but has never had formed vision, it's a big question. We think that actually the wiring would be intact since it would be primed by having the light perception. But could we then get that person to be able to see a face? We don't know, because we don't know if the brain is as malleable as we'd like it to be. But it's something that we feel that we've demonstrated in animal studies is possible. So we're hopeful that it will be effective for other genetic forms of early onset blindness.
One of the questions that comes up, or one of the conversations that takes place a lot within blind communities, is once they were identified as being blind or having light perception whenever they were younger or in their teen years, that they really don't have a lot of eye exams because they don't necessarily have a need to have them evaluated. They don't hurt, they just can't see. And so the question comes up of the discussion really kind of goes around the area of how do you determine if the retinas are non-functioning? I mean, we tend to think of it as visually, if you can't see, then they're not functioning. But what's the test that's actually done, and what's the advice that you guys give out to folks that may have an RPE condition, since that's the one we're talking about today, and how do they go about determining if they might be a candidate?
So one of the answers to that is if you don't get an exam, you'll never know, and you'll never have information like this. So if you had just, in the past, people just sort of threw up their hands and said, what am I doing this for? There's no treatments available, and I just get the same answer over and over again. And to be honest, I'm completely sympathetic to that because, I don't like to burden patients with unnecessary appointments. Everybody has other things they'd rather be doing. However, things have progressed. Things do progress, and over the last 10 to 15 years, for instance, we now have new methods that we combine with a lot of the sort of standard testing that allow us to make better determinations as to whether a person has the potential for vision. We can now actually image the retina with tomography at extremely fine detail, almost like a optical biopsy. So we can tell if the cells are there. They may not be functioning because the biochemistry isn't working. They don't have the proper molecules to capture light, but we know now we can do a very simple, painless, non-invasive test to measure the thickness of the retina or even to see the individual nerve cells. So it's a combination of things that we use to make the determination. It's not just photographs or measuring visual acuity or visual fields. We use a sort of consortium of things from our toolkit to be able to make that determination. And And again, if you check in with the eye doctor periodically, you'll be able to take advantage of that kind of testing and the new technologies that come up.
Yeah, and just to add to that, we find now that we are trying to develop gene therapies for other forms of blinding disease that we're running double time trying to get patients with these diseases to come in so that we can adjust our protocol to measure the sort of responses that would be appropriate for those various conditions. Those are so-called natural history studies. And that's just because people have been told there's nothing you can do and have been lost to follow-up. And one more thing to add, maybe Dr. Maguire wants to comment on this, is for very young children, it's probably not a great idea to measure them too frequently because then they might get scared of going to the eye doctor. And I don't know if you want to comment on that.
Yeah, I think they need a diagnosis, but I'm very much an advocate of not creating phobias because I'm the one who has to take care of the people with the phobias that they develop during childhood. So if you slowly ease people into the examinations and have a good pediatric trained ophthalmologist get the initial diagnosis, that's great, and and then if they can be treated, great. If not, then just sort of check in every periodically to make sure that you're up on the new technology, and if there are any clinical trials that come up, they'll be made available to you.
While we're talking about young kids, one of the questions that we hear within the groups is, how do you guys evaluate? You talked a little bit about the acuity charts. How do you evaluate, and what eye charts do you use for the really young kids?
So I have been more and more impressed over time how sophisticated children are. So this study that led to Luxturna being approved included children from 3 years of age and older. And you would think 3 years of age, they had to do some pretty sophisticated stuff in terms of doing mobility courses at different light levels, and then being able to do vision field testing, et cetera, and by and large, most of the people who came for the study were able to do it and got included. So our youngest was near 3, and theoretically that's a good age to do the treatment. So, but to answer your question specifically, there are many different eye chart systems, some of which don't even require a verbal answer from a child. There's something called preferential looking that a masked observer will look to see if a child looks at a certain side of a chart more than the other, and they can tell the preference is usually gives an indication if they're able to see something. You can also ask a child to have their own card with the letters or figures on it, and they actually will point to them on a card so that they don't have to give a verbal answer. And the very simple thing is a HOTV chart, which use just those 4 letters, what we also call letters optotypes, but only those 4 letters. And most children, if they can't read the entire alphabet, can certainly be taught those 4 letters, and that is a early sort of verbal chart that we use. But the kids are smart, are a lot smarter than they sometimes let on.
Now, are you guys, over the course of time, I think we're talking about 25 years, are you seeing that the research is indicating that the treatment impact is potentially more robust when the treatment is administered in young populations, as young as possible?
Jean?
So our initial results in the animal models definitely showed that there was a much better response in those younger animals than the older ones. And it makes sense because this is also a degenerative condition. So over time, the cells die off and there are less of them left to treat and thus to respond. And so we hypothesized we would see the same thing in humans. And certainly the results of the intervention in individuals of a wide range of ages in humans show that generally the outcomes are people perform at a much higher visual level the younger they are, probably reflecting the number of cells that they have, and this is sort of a generality. However, it will take us more time to analyze all that data in an objective fashion to clearly get a readout. But certainly, one would argue that it would be more advantageous to a person to have vision over a longer span of their life than if they were treated when they were older.
So the burning question for a lot of folks out there that might have low vision or have no vision at this point in their life and have kind of fallen off and not necessarily been to an eye doctor recently, how exactly do you determine if they have the RPE mutated genes that qualify or that has the treatment available now?
It's really quite standard now for people to provide a saliva sample or a blood sample or any other tissue, a skin sample. Typically, it's easiest to give a saliva or a blood sample. And then DNA is isolated from that sample and then screened for presence of mutations in a panel of genes. And sometimes that panel is limited to those that cause early onset blindness, but there are now more than 265 different genes known that when they're mutated will cause blindness. So the testing has gotten really all-inclusive in most cases, screening for a large number of potential gene mutations, which provides even more robust confidence that when you find a given mutation, that is the one. And then typically one confirms that is indeed the disease-causing mutation by testing the parents and seeing if each one of them is a carrier of one of the mutant copies that the child has. And that adds further confirmation to the genetic diagnosis. There are now. Go ahead. I'm sorry.
Go ahead.
There are now many testing laboratories, not only in this country but around the world, and much faster feedback from the test results.
So representing a lot of the parents that are in the LCA group and the kids that are coming up through it as well, we're all pretty well versed and aware that it was the dogs that initiated a lot of this work that has moved through to a clinical trial and now moved into a treatment. So one of the questions that I hear among the kids in our group is, how does the surgery that you're doing on your patients, your human patients that come in today, even though it's an FDA-approved treatment, how does that compare with the surgery method and all of the things that you did with the dogs whenever you were in the research phase?
So the drug is a little bit different because it was modified. It was humanized. Is that the term we use?
Sure. The human cDNA.
Yeah. So dog's DNA is very, very close. The gene is almost exactly the same, but not quite exactly. So when we were testing the original dogs, it was more canine-based in terms of the gene that was introduced. So the surgery is essentially the same. You take a small tube, place it under the retina, and inject the medicine. The actual mechanics are a lot different because imagine, dogs have their eyes on the side of their head. They have this big snout in front of them, which would, if you were in a human operating theater, that would hit the microscope. That would be a bit of a problem. So a lot of it was just little sort of mechanical things to properly position the animals and just compensate for different morphology, different look to the face and to the snout. But when it came down to the actual treatment, they had a injection under their retinas exactly like the humans, and they respond in an amazingly similar way too.
Yeah, I'd like to add that two of those initially treated dogs are now living at our house. At the end of the experiment, when we had all of our data, we decided they had given us such great information and been so valuable, and we loved them in addition, so we actually went all the way to the provost at the university to be able to adopt them and give them a good home.
You guys, can you share with everybody out there, especially the kids that are listening, what are the dogs' names? And what type of dogs are they?
So it's Mercury, Mercury and Venus. Venus is the mother. Mercury is the son, and they are Swedish Briards that were bred by Christina Narstrom, who's a Swedish veterinarian, and they are very unusual. You can Google image Swedish Briards. In fact, I think their pictures are probably going to come up. But they're very unusual and very beautiful looking dogs, and they run around the backyard and they look, bark, and move around just like any other dog I've ever had.
And they're very, very sweet. If any child listening would like to meet them, they're always welcome to meet them in Philadelphia.
Now, a curious question, just as dog owners out there, once they received the treatment, you had mentioned that they had the same type of rapid pickup. Did you find that there were odd little things that you kind of had to reteach them, as we were talking about with Creed and some of the other things that we're seeing go on?
Yes. So these animals were born and raised in an animal facility, which is on one floor, and there are no windows, no grass. So the first time that I got approval to take the dogs home, I brought them outside. They saw grass, and they're pawing it because they'd never seen anything with that texture or shape, color before. And then stairs were a new phenomenon for them. They'd never seen stairs, and they had to kind of figure out, this is how you walk downstairs. Now you'd never know there was a problem, but that they had never had that experience, and it's not a problem for them because they can see the stairs, but it was a new experience.